Thanks everyone for coming. Thanks to Kim and Mark as well for putting up with me for another year. So, do I need, am I not loud enough? Low?
No,
Testing.
you're not on that.
All right.
You're here.
I messed everything up, man.
That's all right.
Cool. So today we're gonna. So a couple years ago, when I came for the first time, I talked about cannabis genetics and why they were important to the crossover Canna pharmaceutical kind of industry. Today we're going to talk a little bit more about that stuff. We're going to and learn, maybe a bit optimistic, right? We got a lot of stuff to cover today. Some of the stuff we're going to touch on today are listed here. But at the end of the day, what I really want to come away with is thinking about cannabis in a little bit different way, right? What I'm going to cover today is actually a continuation of what Ethan was just talking about. Ironically, he's always one-upping me. So we're going to talk about synergy, right? And why understanding cannabis genetics is really so important to being successful moving forward, right? So we're going to start a little bit about the role of cannabis in pharmacology. So 80% of the world started using medicines that were plant-derived and still to this day do. 20% of the world uses what we know as the traditional Western medicine, pharmaceuticals, pills, elixirs, that kind of stuff, right? But they're not mutually exclusive, right? Today, 40% of all pharmaceuticals are still plant-derived, including 20 of the top most used pharmaceuticals today, right? So what we're talking about here isn't really new, right? And it's so not new that in 79 AD, Pliny the Elder said that you could use the roots. In 90 AD, I'm not even going to try to pronounce his name because I'm going to screw it up, right? But same kind of thing. J. Russell Reynolds, who it was probably better known for being Queen Victoria's court physician, published in 1859 that there was no better medicine available than cannabis. These were taken, by the way, I was not the originator of these. I didn't find them. These were all stolen from Dr. Meshulam in 2016 at the Canada conference, and I've used them ever since, right? Because it's not that we don't know, it's that we have forgotten, right? Okay, so let's talk a little bit about cannabis and why it's important, right? So as we learn, trichomes, leaves, roots, plant latex all contain important pharmacological compounds, right? And I'm going to disagree with Ethan a little bit in that what we're finding now is that in the roots and other parts, there are a lot of other compounds that we did not know about, including cannabisins and cannasative. It's an alkaloid. I'm blanking on the name right now, but along those lines, my team has done a lot of transcriptome analysis. We're a genetics company, and so we look at expression of the genes. And what we found is that 50% or almost 50% of the expression in any trichome is not cannabinoid or terpene. It's alkaloid, right? So there's a whole other class of compounds being produced in the very same trichome that we're talking about extracting, right? That we haven't even begun to understand, right? And again, highest concentration of plants. Some of these things you're going to hear over and over in these talks today, I think. Highest concentration of ingredients are found predominantly in female plants, except for those that are in the roots. All right, so why are we really concerned here, right? Or why are we really here, right? We've already discussed there's hundreds of compounds, right? Now, the majority of these compounds have a function in either receptor kinetics, right? Or other biochemical pathways in a non-receptor mediated fashion, right? So we'll go into that a little bit more, right? The majority of these things have already been studied, terpenes at least, and almost every terpene has GRAS status. That's an important consideration, right? So GRAS means generally regarded as safe. The other thing is that as a phytochemical agricultural tool or API raw material, right? You can get a lot of compounds from the dried plant, or as Ethan said, from the fresh frozen plant. Okay, this is just to show the various ways that you can consume cannabis. And the reason why I have this up here is because every one of them gives you a little bit different chemistry set. Raw is acidic. We'll go over some of these more, but topical. I want everybody to pay particular attention to the topical application. The reason being, as we will cover in a little while, is there are a bunch of terpenes that are natural skin penetrants and have actually been studied so much that we actually have what's called effector ratios, right? On how they enhance skin penetration, right? So we can actually look at, as I said, the synergy, right? There's a lot of compounds that have evolved together to produce an effect, and taking them apart may not be in our best interest, right? So sublinguals, again, under the tongue, if you have something that's highly bioavailable, that can penetrate the blood-brain, any kind of membrane barrier, we'll talk about that in a little bit as well. Smoking, vaporization, more or less the same. One doesn't give you lung cancer, that's great. So, and ingestion, right? So ingestion is an important consideration as well, right? Because now we have a different chemistry, right? Because now it's not going directly from the blood into the brain. It's now being processed by the liver and giving you a metabolite that affects you completely different, right? So not only do you have 3 different medicines in the plant, depending on how you use those medicines, you can get even further differential effects. Okay. Another quote that I find very interesting, right? So 1464, a sheik was forced to give his son hashish to control epilepsy. Wow, what does that sound like? All right, so again, the downside is he was forever had to be taking that product. How is that any different than today's pharmaceuticals? What pharmaceutical do we take today that cures us that we don't have to take anymore? So let's talk about medical cannabis and the different types of medicine, or any cannabis for that matter, right? So raw, you get one set of compounds, right? The acidics, by the way, the plant does not get you high, right? Can never get you high. It's a man-made function that gets you high. You have to heat that product. That does not occur in nature. Also, aging bypasses this, right? So CBN, CBL, those breakdown products that are not heated are also not psychoactive. So when you look at cannabis, you have to actually understand what you're going after. Non-receptor mediated, receptor mediated, and unknown, right? So we don't know. These aren't really well studied yet because there's not a lot of them, right? So once again, right, you have to really understand the plant and what you're trying to get out of it, and that's where some of the genetics comes in, right? By the way, every one of these, I thought I was going to have a pointer, but never mind. But I'm not supposed to move around too much. But every one of these in the boxes by KCCBD, those are all peer-reviewed effects, right? These are not anecdotal. Every one of these boxes contains information that was taken from a peer-reviewed scientific or medical article. Okay, so now let's talk about cannabinoids and what we're really seeing in the science today, right? So recent publications, how CBD and cannabinoids can cross the blood-brain barrier, right? Well, if you take a look at what's highlighted here, you'll see a couple of key words, right? Inflammatory cytokines. Well, that's the big buzz today with COVID, right? Some of the data today is showing that CBD actually may help reduce that cytokine storm, right? On the bottom, diseases such as epilepsy and schizophrenia are also effectively treated, not necessarily by the cannabinoids by themselves, but in addition to some other medication because of the repair to the blood-brain barrier and the penetrance across the blood-brain barrier, right? So these are important considerations, right? And they're all tie up as we keep going, right? We got neuroprotection again, at least a well supported, right, effect of cannabinoids, right? But here we have Alzheimer's, immunoregulatory role of cannabinoids during infectious disease, again, pointing towards the use of both cannabinoids and terpenes in our treatment of a lot of diseases like COVID, SARS, some other things. In fact, there are some terpenes that are so good at killing bacteria that they can't be established for assays, right? Listeria cannot exist in the presence of any cannabinoid or terpene. So this is probably what everybody is more familiar with, the use of cancers or the studies of cancers. This is a paper that was published recently, 2020. I recommend everybody take a look at this, right? Because not only do they talk about cannabinoids, but they also go through terpenes, right, and flavonoids, and they cover the effects that have been seen, but they also talk about which one of these things is currently participating in clinical trials. As you can see, there's no clinical trial for THC, but if you look at some of the other cannabinoids, you would see a list of clinical trials for CBD, CBC, a whole bunch of them, right? So what we think is not happening is happening in a lot of places, just not in the United States. All right, so we've talked yesterday and today about the cannabinoids mainly because that's what everybody thinks of cannabis as, right? And clearly THC, CBD, all of these cannabinoids. We're going to spend a little time talking about a fair amount of time talking about terpenes today, and I'm going to go over flavonoids a little, only because there's only a little information available about flavonoids in cannabis, right? So we'll cover that a little bit, and maybe next year I'll get invited back and I'll talk about flavonoids. So. All right, so let's talk about the medical uses of terpenes now, right? And this is all, again, peer-reviewed articles. So linalool, which I will also disagree with Ethan a little bit on. Linalool is in vanishingly small quantities in certain varieties. But as you'll see, in other varieties, again, determined by genetics, it's not that, small a contributor, right? So, but importantly, right, we see that linalool, which is a tertiary substituted monoterpene, right? It's an alcohol, does two different things. It's an anti-anxiety and it's a sedative. Well, those two things kind of go together, right? If you're not anxious, you'll be able to sleep better, vice versa, right? So we have, again, those were mouse models, right? We have alpha and beta pinene, right? Which are now recognized to be anti-malarial, antibacterial, anti-tumor, gastric protective, anti-anxiety. Interestingly enough, there's another secret sauce piece of the pinenes, right? And some of you may be familiar with a variety called Blue Dream, right? Blue Dream is one of the few varieties that has a reversed alpha pinene to beta pinene level, right? Beta pinene being higher than alpha pinene, very rare in cannabis. Ironically, beta pinene and alpha pinene play differently in men and women's brains, which is one of the reasons why women choose preferentially Blue Dream every time they run the poll. It's been run 3 times, and every single time women pick Blue Dream preferentially, right? So now we can start to understand why some of the things happen, right? So there's a different brain wave, and if we look at some other stuff here, right? So limonene and terpinolene, right? So here we have direct evidence of how these things are affecting your actual brain wave, right? Terpinolene, focus, limonene, right? Gives you gives you that more relaxed feeling, right? All right, so came out a little weird. The beta caryophyllene is actually for the systemic review of essential oils. We talked about that a little bit. Beta caryophyllene is in fact a wound promoter, a wound healing promoter, right? It moves, various cells to a wound site to help promote healing, right? Limonene, as Ethan said, anti-anxiety, right? And then, again, I had mentioned earlier, transdermal drug delivery, right? Well, enhanced by, and this is the paper where it lists all the effects ratios of all the different terpenes, right? So I'm trying to give you guys a roadmap to why we think in my company the way we think about this plant. Okay, so we covered a little bit. This is a brief review, right? Terpenoid literature, this is what you'll find, right? All of these things have been definitively shown in peer-reviewed journals, right? In both animal and mammal, human model, right? So pain blockers, anti-inflammatory, right? ACH inhibitor. So that's particularly important. Ethan talked about alpha-pinene, right? Alpha-pinene helps prevent memory loss. Well, it does it because it's an ACH inhibitor. ACH inhibitors also elevate mood and prevent, memory loss. So now we have an understanding of how these things are actually doing their work. And to answer your question, these are some of the reasons why we do focus on the terpenes, because they are acting in synergy, right? And they do have effects that, when combined together, are equal to greater than the sum of their parts. All right, we're going to jump into flavonoids, and we'll go back to terpenes. So flavonoids. This is a list of flavonoids that you will commonly find in cannabis, right? These were not isolated from cannabis. This is an old paper, but you can see flavonoids already have some medical benefit that has been studied, again, peer-reviewed. So let's talk about orientin in particular. Orientin seems to be this miracle flavonoid that's in everything. It's in the cosmetics, it's in nutraceuticals, it's everywhere, right? And it does all these things: anti-inflammatory, antibacterial, anti-aging, okay, whatever, right? Neuroprotective, radiation protective. That's pretty interesting, right? So now when somebody asks you, wow, why do you study cannabis? Well, it's because the thing is chock full of good stuff, right? It really does. And some of these things, when you look at where you would get some of these compounds from other organisms, right? Cannabis is actually producing levels that would be competitive in that extraction market, right? So not everything, right? But some things. Let's see here. All right, so these are cannabis-specific flavonoids, right? So the only place you find cannaflavins is in cannabis, right? We've already found that it helps with Alzheimer's, right? Pancreatic cancer and other types of neuroprotection, right? So what I didn't include here is right now there's a tremendous amount of work being done in Israel on the actual anti-tumor properties of cannaflavin A and cannaflavin B. So again, unfortunately, the U.S. seems to be a little bit behind in some of this research, but if you look deep enough, it's there, and it's telling us where we should be looking, right, in the United States. Okay, so I'm going to go back to terpenes now, and we're going to talk a little bit about receptor action and allosteric interaction, right? So this is the meat of how terpenes are doing some of their stuff, right? So what we'll see here is that terpenes actually do act at the GABA A receptor, right? They do have ACH inhibitor activity, right? And we talked about how the olfactory stimulus earlier, right? The brainwave focus for terpinolene, right? And then I'm forgetting the other one that was on there that actually had the low power. I think it was limonene, actually. Yeah, it was limonene, right? So we're going to go over a concept of terpene claves. Now, let me explain this a little bit, right? So when you look at the terpene profile, and Jeff will probably be talking about this in a little while at some point. When you look at the terpene profile of a cannabis plant, what you see is that there's 3 classes of terpene concentrations, right? You would say concentrations in air quotes, right? You have the primary terpenes, which represent the predominant production of terpene in that plant. You'll have a secondary level of terpenes, which are typically present 5 to 10 times lower. And then you'll have a really low abundance set of terpenes, right? In that primary set of terpenes, what you'll find is there are 6 major terpenes that produce in combination 7 distinct classes, right? Beta-myrcene is one of the predominant terpenes. Beta-caryophyllene, limonene, beta-farnesene, terpinolene, alpha-pinene, right? And each of these predominant terpenes has its primary effect, okay? So this is what's going to establish the overall direction of the therapeutic, right? So, and beta-myrcene, I have to say this, everybody gives it a bad rap. It is, in fact, not the reason why you get couch lock, right? It is not what makes you a couch potato, right? So it's what it's in combination with, right? So beta-myrcene can be codominant with alpha-pinene, and you're not going to fall asleep with that one, right? But if you have beta-myrcene codominant with limonene, guess what you're doing that night, right? So it's important, again, right, to understand how these things play together, because now we look at the secondary terpenes. The secondary terpenes, which are the ones that are 5 to 10-fold less in concentration, actually contain a group of compounds that will, in fact, do two things: smell more than the primary ones, right? The odor ratio between limonene and myrcene is 100 to 1. So basically, you can have 100 times less limonene and smell it more than myrcene, okay? The other thing that they do is they frequently have as high a contribution to the effect that you get as the primary terpene. So when you look at things like linalool, even though it is there low, it may in fact be the primary influencer of that therapeutic in that cannabis variety. Right? So it's important to not associate the primary terpene with what you're getting. And it's important to understand that you need to look at all of the terpenes to understand what you're really dealing with. Okay, so let's take a look at this a different way. So I covered the concept of the 6 or the 7 clades, right? So this is proprietary, so all you guys get to see is clade 1 through 7, right? But if you take a look at the different clades, not every clade is going to give you the same level of anti-nociception, right? So this is where the genetics come into play. If you aren't going after the right genetic with the right compound, you may be wasting your time, right? So, okay, so brief summary, right? Flower therapeutics, as I just said, are largely determined by the terpene, right? And not necessarily the primary terpene. And we know about all these terpene effects in the absence of cannabinoids. So everything I just talked about is well studied and has nothing to do with any cannabinoid. Okay, so agriculture. Ironically, we've spent all this time talking about how cannabis affects us, and cannabis didn't do this for us at all, right? Cannabis is, in fact, one of the most flexible plants. It is one of the few plants that actually colonizes almost everywhere humans does. There is not, well, no, I'm not going to say there's not another single plant, but there are very few other plants that have that ability, right? And that's because of the plasticity of the genome, of what it carries with it to do what it does, right? Through this section, we're going to talk about a little bit how those compounds really evolved and what they're really there for, right? So terpenes, as it turns out, are there to do environmental reconstruction, right? So what you will see is that terpenes will either act as a protectant, they'll act as an attractant, they'll act an insect protectant, they'll act as an insulation or solar radiation protectant, right? So the structure of these things, of all the different terpenes, does a lot of work in free radical scavenging, turning things on, turning things off inside the plant, and turning things on and turning things off inside insects as well, right? Guaiol, insecticidal terpene. So molecular targets for compounds and essential oils. This I highly recommend because this tells us really what the things that we're talking about in terms of terpenes were really designed for, right? Like, what do they do? And as it turns out, there's a whole range of terpenes that mimic an amine, right? So octapamines, which in humans are norepinephrine, right? There's a norepinephrine equivalent in insects, right? All of these things are affected, right, by various tertiary substituted monoterpene alcohols, right? So on the left here, on the right, it's the other left. On the right, you'll see what are octapamines that are isolated from either humans or insects, right? On the left here, you can see all of the different terpenes, and just by looking at the terpene structures and those structures on the right, you can see how some of them might in fact mimic or fool a receptor. So, as it turns out, linalool and other alcohols are the ones that have the most effect in the octapamine receptor and cause all sorts of bad things to happen in insects, including oviposition deterrence, contact killing, right? attraction of your most hated predator that will eat you. All of those things are controlled by these and are very effective in agriculture. All right, so how can you use this to your advantage, right? Well, we'll go over that in a second. But so here we talk about terpenes modulated octamy receptors, right? HIPV, herbivore-induced plant volatile. So when the deer comes and eats, as Ethan said, there's a whole response reaction where monoterpenes and some sesquiterpenes, which are bitter, right, and make you not want to eat it, right, are kicked into high gear, right, to help protect that plant, right? Certain terpenes are also only in roots. That's important because they condition the soil to be able to make that plant survive a harsh environment, right? So all of these things are different functions that terpenes developed. And it just so happens that in some cases, some of those functions have an effect on us as well, right? Through some of our second messenger systems and whatnot, right? All right, so genetics. Because this is about money, it's always about money. We're going to talk a little bit about API and maximizing the revenue, right? So what I've done here is Cannaflavin A, Orientin. All I did is because I run a chemistry department as well, right? So I have to buy standards. So I looked at how much I'm paying for these standards, right? Now, granted, this is at milligram quantities. If you were to go and buy these things in bulk, you'd probably get them closer to $1, a milligram, right? If you were buying large quantity, right? But if you think about it, $1 a milligram, right, is still a hell of a lot of money, right? Like it's $1,000 a gram for something. That's more than gold, right? So, so we are looking at potentially compounds that have fairly high value in the API market, right? So some KPIs to think about as we continue to talk, right? If you see, right, so oil cannabis is generally planted 1,000, 2,000 plants per acre, right? Conservative, real conservative, because there are guys up in Humboldt County that are getting 10 pounds a plant, right? So conservative, 1 to 3 pounds per plant, right? This is the important number. 100 ppm on a COA, right, is equivalent to about 4 grams of raw material per 100 pounds of starting material, right? Keep that in mind. All right, so this is a typical flavonoid COA, and I'm putting it up here for example, right? So what we're looking at here is a spread of flavonoids, and you can do this with terpenes, you can do this with cannabinoids as well. But what you're looking at here is if I'm planning on extracting, is this a good plant to extract? Right? Well, for Cannaflavin B, not so much. Cannaflavin A, well, pretty much. But look at Orientin. So now I'm going to be honest here. This was not a standard plant. This was after a stress treatment. We were looking to see if we can induce flavonoid expression after stress. Right. So yeah, the Orientin number here at 551 and 1637, that's a little artificial, but think about that. So this is 16 tons, 4 grams per acre, right? So when you go to determine what's a good plant to extract, this is some of the stuff that you're looking at, and some of the points you need to keep in mind, right? So again, if you look at the right side, we actually go through and do the math on that conservative estimate, 200 grams of Cannaflavin A per acre, right? At $1 a gram, right? 200 grams times 1,000. That now you're up in the $200,000 per acre, at wholesale cost, right? That's the wholesale cost if you can get $1 a milligram for Cannaflavin A. The nice thing is that you also get Cannaflavin B in many cases, and you'll get a bunch of other flavonoids. You'll also get terpenes and cannabinoids, right? So suddenly, when you look at cannabis as an API, right, and you look at the scale on which you can do it and the price, suddenly cannabis becomes a very attractive plant to get some of these medically important compounds, right? And again, the key to getting the most value of your money is to get the right genetics. We're going to explain why, right? We just talked about that stuff. All right, so the kind of work that I do involves lots of screening. And so this is an example where this is the whole experiment here, right? Where we did a whole bunch of stuff, looked at a whole bunch of plants, and you can clearly see not every plant is going to be a good extraction material, right? Some of them have great ratios of Canna A and B. Some of them have reverse ratios, you can see, right? Where there's a 1 up here, where Canna and B is actually here, number 3. So the ratio of Canna and B to Canna and A is actually reversed, right? So these are all genetic examples of why you really understand what you're looking at. This is another one, and this kind of closes the whole talk. What we're looking at here is a number of different proprietary varieties that my team and I have bred, and you can look at the extent of terpene profile. The important thing to understand here is, in fact, the synergy, right? Beta-myrcene and alpha-pinene, right, up there. If you look at the rest of the terpenes, you will find cannabis is not confused. You will never find a cannabis plant that has a primary uplifting terpene mixed with a bunch of sedative terpenes. Does not happen in cannabis, right? Even when we have such hybridization and messed up genetics, the plant still knows how to figure it out, because at the end of the day, these things are responding to environmental signals, right? And they still produce what they need to survive, right? So again, understanding the genetics and understanding the cultivation is how you're going to maximize your API, right? Conclusions, what we learned today, right? So it lives everywhere. Not many plants do this. Because it lives everywhere, it's developed a unique chemistry set. Those chemistry sets do all sorts of stuff in the environment, condition the biome to allow it to live. Some of those effects have been studied in humans quite well with a lot of data, right? And it's important to understand that you cannot get everything from every cannabis, right? So that's where understanding genetics really comes into play, right? Because if you go after the wrong genetics, you won't get what you're looking at all, right? And at the end of the day, because of the synergies we talked about, the ability to have the terpenes that penetrate skin, right? And so you can take your keef that has a high level of linalool or alpha-disolvol, and you can just rub it on your skin, and that will be an effective treatment, because you have, in the synergy of that plant's chemistry, what you need to get across the skin. And I think that's it. Oh, yeah, one last thing. This is phytochemical agriculture. It's been going on for hundreds of years. Cannabis is a plant like any other, right? It's time to start thinking about it that way, so that we can do the correct phytochemical agricultural, building and treat them like the factories that they are. And thank you very much. My team, everybody involved, thank you, and some of what we do.